Hey, what's going on?
Not much. How are you?
Going well, going well.
Good.
Yeah, I wanted to let you know, I've been doing cars lately, and I'm
Uh-huh.
completely transformed how I've been doing since the last time we
Oh, yeah.
uh I guess I just wasn't like, I don't know, I wasn't really, I was kind of just reading the passage just for reading's sake, and I wasn't really asking myself questions actively, and I found that that was like made the biggest difference.
Nice.
Yeah. So I'll let you know that. But yeah, um I wanted to go over like a specific uh passage in bio and then just like briefly go over some like psych-social stuff.
Okay.
Yeah, so I'm gonna share my screen. Okay. You can see that?
Mhm.
Okay. So I wanted to just like go through the passages the way that I like would on like test day. Because the last time we spoke a lot about like visualizing and like kind of mapping out like how my thought process is.
Mhm.
And so I wanted to go over this one specifically because I kind of had a hard time going through it.
Yeah.
So maybe you can help me with correcting some of my mistakes here. Um, so I mean, I can just like go through this the way that I normally would, or would you prefer that we kind of go through it slowly? What do you think?
Um, yeah, I think going through it slowly would be best because that's pretty much what you should be doing anyway. It's just that when we start having you take practice tests, you're just gonna work on being faster, but I guess like, practicing doing it slowly will let you do it smoothly, and then doing it smoothly will let you do it fast. So yeah, I would say to go through it slowly and kind of like tell me what they're being what's being said in each, I guess, uh set, and look out for those buzzwords as well.
Okay. So beginning off the first paragraph, so digoxin is a cardiac glycoside. So I'm saying cardiac, and I'm already thinking like, okay, so it's involved in the heart somehow.
Anything
Um, Say again?
anything else there?
Um, I mean glycoside.
Yeah. What's that?
I mean I'm thinking like sugar, but I don't know the specific like structure.
Yep. So yeah, it'll be talking about a sugar here. So like the cardiac part is like kind of less
Okay.
something that they would ask about because they usually start off saying something like about some clinical relevance. Um, but the glycoside, like, yeah, they could ask you about like glycosidic bonds and stuff, and that's the type of bonds that exist in sugars. So, uh, yeah.
So this has a positive inotropic effect, which is increases the force of contraction. So I'll
Yeah.
probably highlight that just for memory. On heart muscle by inhibiting the sodium-potassium ATPase on the myocardial cell membrane. So I'm thinking about the ATP pump essentially,
Okay.
um, and says that digoxin is inhibiting this pump essentially from.
Hmm. So, uh, so did I don't, I guess for the last passage that we did, or the passage that we did on BB, I didn't go over like writing out proportionalities, but that's going to be useful for most of the time. So I'll write one out. Let me just, I'm gonna clear this. I'm gonna do that. Then so I would say that digoxin, and if something the symbol for proportionality is this,
Okay. Mhm.
And if something's like proportional, it means like if something, so like if I write like A is proportional to B and I plotted it like this, it would be like a straight line like that, right?
Mhm.
Now, if it was this, like an exponential, it would look like, oops, it would look like that, right?
Yeah.
Now, in inverse proportion, we write that like this. If it's an inverse, it'll be proportional to 1 over B. And what do you think this graph would look like?
I mean, it would essentially be straight line, but going down.
Like that? Like that?
Yeah.
So let's see, right? Let's see if we can like plug in numbers. So, when A is 1, 1 over B would be 1 over 1 or 1. When A is 2, 1 over B would be 1 over 2. When A is 3, what would this be?
Sorry, it's like thundering out here. Can you repeat that?
Sure. So we can try plugging in values. So if we plug in A is equal to 1, and then we plug it into 1 over B, it would be 1 over 1, which is still 1. But when A is 2, 1 over B would be 1 over 2, right?
Mhm.
Which is 0.5. And when A is 3, what would 1 over B be?
0.33 or 1/3.
1 over 3, yep. And let's go like kind of skip forward. Let's say like, what about 3,000?
It'd be 1/3 times 10 to the negative.
1 over 3,000, right?
Yeah.
So as A increases, what happens to B or 1 over B?
It's going down.
All right. And it seems like it gets smaller and smaller and smaller, but is there any like end to that?
No, it goes to infinity. Oh, okay, so instead of it being a straight line down, it should be like should kind of be yeah, the other way.
Exactly. Because what number can B not be?
Say it again.
Oh, so what number can B not be?
Whatever A is.
Well, so think about what number.
There's zero, is that what you're talking
Yeah,
about? Okay.
yeah, because 1 over 0 would be undefined, right?
Right.
So that can never be 0. So what that means is that I'll draw like this thing again. So do you remember what asymptotes were? What's up?
I'm trying to think back to
So an asymptote is like an imaginary line. It's usually drawn as a dotted line that something can infinitely approach but never touch.
Mhm.
So you actually see it in in Michaelis-Menten enzyme kinetics, where you have substrate concentration as your x-axis and rate or v as your y-axis. You have a dotted line here, right, for v max,
Mhm.
and the enzyme infinitely approaches v max but never touches it. So you're familiar with this, right, in
Yeah.
Michaelis-Menten. Yeah. So this Vmax dotted line is an asymptote, which means that this actual line or curve infinitely approaches it but never touches it.
Gotcha.
So when it comes to an inverse proportion here, the axes are considered to be asymptotes, which is why the graph of it would look like this. That type of like hyperbolic shape. So this is really something that they like to test. I've seen it a lot. And it's because most people would think that, if a proportional linear relationship is like this, then, you know, inverse would just be going down. But that's where, you know, the special nature of the inverse proportion comes in, where yeah, it can't be zero. So all right. So yeah. So also like I see like a lot of tons of tutors and stuff write proportionalities like this. Like if it's a direct proportion, they would write like A goes up, B goes up, some arrow stuff like that.
Mhm.
That's not a good way of doing it, because that doesn't give you any insight about the actual mathematical relationship that lets you plot plot it on a graph. But yeah, any questions about that?
No, that makes sense. I mean, I usually do the arrow thing because
Mhm.
it's just like, I guess it's just easier by default to just, okay,
Mhm.
but okay.
Yeah. So
That
yeah,
makes sense.
I would still say yeah, definitely use so for um so okay, so for passages like BB, like the last BB passage we did was probably like one of the few that I've seen where we didn't use proportionality, but mostly all the time you're going to be using you want to be using proportionalities, because there's often going to be so many acronyms to keep track of. So but okay, so we know here that digoxin inhibits this ATPase, right?
Yeah.
So would I write the relationship between digoxin and this ATPase as a proportional one or inverse proportion?
Inverse proportion.
Yep. So then I would just do 1 over um, sodium potassium ATPase to show the inverse um, proportion. Now, what else can you tell me about this pump?
It's so it has to do with creating like a proton gradient. Or it's usually found in like depolarization, action potential stuff. So 3 sodiums coming or going out, 2 potassiums coming in. Um,
Yeah, so tell me a little bit more about what you said. So, because you said like depolarization and stuff. So, yeah, tell me a little bit more.
so the first thing I'm thinking about is just like ion concentrations, essentially.
Okay. And go on with that, what you're saying about that.
And um, ATP is just pumped through. Actually, I'm not. Is the ATP separate from the ATP pump, or is it the same? These are the same. Okay. Yeah.
No, they are separate.
Oh, they're separate. Okay.
Yeah, because when you say ATP pump, you're talking about the one in the inner mitochondrial membrane, right?
Yeah.
Oh yeah, so yeah, that is different. Uh, so what the sodium potassium ATPase does is, well, okay, so you mentioned depolarization. Tell me a little bit about that. Like like, yeah, tell me about that, about what that is.
So change in cell membrane potential. Um, I guess another aspect is, so I guess I'm thinking of like voltage. So like,
So
what?
like, let's say this is the um so what would be the so yeah, okay. Tell me like I guess I still say I'm still kind of asking you this vague thing, but tell me like about what this like the state of the cell or let's say like neuron or something, right? Before depolarization.
Before depolarization, I guess like uh voltage would be like below the threshold, I guess.
Okay. What do you remember like what numerical value that might be related to or anything like that?
Be was it -70?
All right, good. Now, why? Why is it at -70?
Because there are more potassium channels open.
So let's see. Um,
So as it relates to like it's closing, that's more
so
applicable.
so let's think about okay, so is there a higher amount of
There's more negative charge inside the cell, I guess maybe
yeah.
is
So why isn't equilibrium established? Why doesn't enough sodium, leave the cell or enter the cell? Why doesn't enough like potassium, leave the cell or enter the cell to be at equilibrium?
because it takes energy to for them to move against their gradients.
So that would be if we moved. So like if this is like, let's say a cell and this is so we have high sodium outside and low potassium outside, and then low sodium inside and high potassium inside. So when a sodium pump opens, sodium, does it go from the outside to inside or inside to outside?
Goes from inside to outside.
So where would it, so
Where
diffusion,
would it originally be?
so diffusion would go from high to low, right?
Mhm.
So so if a sodium pump opened, would the sodium go like in or out? If there's high concentration on the outside.
That would go in.
Yeah, go in. And we have high potassium on the inside, so that should go out, right?
Mhm.
And that's what should happen, right? That's what happens. Diffusion doesn't require energy, and that's what should happen. But the reason it doesn't happen is because of the sodium potassium ATPase. That uses ATP, it uses energy to pump out three uh sodiums and take in two potassiums. So we have a net loss of positive charge, which is why it can be a negative charge on the inside. So
Okay.
that's how you get your -70, right?
Yeah.
Because it things should be at equilibrium, but the sodium potassium ATPase, also known as like the sodium potassium pump, uses energy to create that concentration gradient. Okay. So that's what the sodium potassium pump does. It forces a concentration gradient to exist. Whereas without it, uh everything should just be at equilibrium. That kind of makes sense.
Yeah. Okay.
Okay. Yeah, and it's different from what you like the ATP pump that's like, in the mitochondrial, yeah. Um, okay, cool. So yeah, so far glycoside, I would say the buzzword so far are glycoside and ATPase, the sodium-potassium ATPase.
Okay. Digoxin is not effective in all patients with congestive heart failure because it can be inactivated and excreted as the metabolite dihydroxy digoxin or dihydro digoxin.
Okay. So that's the name of the inactive form of it, right? With a dihydro, which could mean it's attached to like some type of maybe two water molecules or something. But yeah, you can, yeah, continue.
I guess at this point I would probably like write out. So I think my like first instinct would be to let me see if I can write on this. There we go. Okay, so I would probably um dig um I guess so like whenever I see inhibits, I should just think of inverse proportion. Is that kind of what you're getting at then?
Uh, yeah, I mean that would work. You know,
Okay.
uh, yeah.
Okay. I think also before I consider doing it that way, usually what I would do is probably say like, okay, well, it's inhibits this pH,
Oh,
and then
so if you're gonna do it with an arrow, like
mhm,
I thought you were gonna say like, the thing where if this, like let's say digoxin goes up, the ATPase goes down. I thought you were gonna say something like that. But if you're using an arrow in a pathway way, like a pathway, then the proper like notation would be if it's inhibiting like this flat.
okay, fair enough.
You know what I mean?
Yeah.
But but yeah, yeah, I would definitely either like, if you're writing it like a pathway like this or like the proportionality like this.
Mhm.
But I think yeah. So for this, so far, that's what we essentially have. Um, what I would also add though would be that like gut microbiota is proportional to dihydrodigoxin, which is inversely proportional to digoxin. So you could see how you can chain these proportionalities together and, you know, relate them in that way instead of just putting a bunch of arrows and stuff. Like if you put like up here and down here,
Yeah.
like that's not ideal. But uh but yeah, you could see that the presence of gut microbiota would be proportional to the dihydrodigoxin, which is the inactive form. So dihydrodigoxin would be inversely proportional to the actual digoxin. Yep.
Okay. Okay. So next passage or next paragraph. So I'm just gonna call this E lenta. E lenta, a gram-positive bacillus can reduce digoxin, although high levels of arginine inhibit this reaction.
Okay, so we got some more information here. Now, tell me if we have a buzzword here.
Gram-positive.
Yeah, good. Tell me about that.
So it's relates to bacteria.
All right.
Uh, gram-positive has like an extra um let me make sure I say this correctly. There's a difference between gram-positive, gram-negative.
Mhm.
Gram-positive has the extra peptidoglycan layer.
So
Oh, it has the extra like just cell layer. Gram-negative has the peptidoglycan.
So if you so the gram positive, it's named that because it takes up a dye, and if you remember that the cell wall of a bacteria is what picks up the dye, then the one that has a really thick cell wall will pick up more of the dye. So for gram positive, that would be you have a cell membrane and a really thick peptidoglycan cell wall that picks up the dye, which would give it a purple color. Whereas gram negative would have that cell membrane a well, I'm going to just make this thicker by comparison. But the gram negative will have a really thin peptidoglycan cell wall, but it will also have an outer cell membrane. And the thin cell wall means it does not pick up the stain as well as the gram positive. So instead of purple, it would be like kind of pink.
Yeah.
But does this gram negative one seem familiar in a way? Do you like have we seen anything that has perhaps two cell membranes like that? Or two membranes?
Not ringing any bells.
What about
Oh, is it like viral abscess? Or
oh
no?
oh no, what about like mitochondria,
Okay.
right? Because mitochondria has like an outer membrane and then an intramembrane space and then an inner membrane and then the mitochondrial matrix, right?
Yeah. And it's similar to bacteria.
Yep. Do you know like the endosymbiotic theory?
Um,
Like basically the idea is way back in the day. Um
Off of eukaryotes, is that what you're
yeah. Like mitochondria was some ancestral prokaryote that got taken in by some other type of cell, and that's kind of how, since they're symbiotic, that's how eukaryotic cells, or particularly like the organelles of the membrane-bound organelles of a eukaryotic cell, that's how they came to be. That's also why mitochondria have their own DNA, right?
Mhm.
Like, have you heard of like mitochondrial DNA?
Yeah.
Yeah. That's like something you pick up from your mother because from your father, you're really only getting the nucleus of the sperm cell. But from the mother, you get everything else. You get the rest of the cell plasma, all the other stuff. So, yep. So yeah, good. So that's the difference between gram positive and gram negative. And they're saying that this gram negative egg thing um, can reduce digoxin and, well, sometimes it can be confusing seeing the word reduce. Um, why do you think?
Because it's not quite the same as inhibit.
Oh, I was gonna say because it, at least for me, I'm thinking, does this mean it's like redox reduce or make something less of, right? So I don't know what they mean right now. Um, because if it's redox reduce, then maybe that's how it can turn it into dihydrodigoxin. But
Mhm.
it might. But I guess it like I'm gonna say that maybe it's saying to reduce the amount of digoxin. So I'm gonna go with that for now. So I'll write egg is inversely proportional to digoxin, 1 over dig. Although high levels of arginine inhibit this reaction. So maybe I would say like high arginine, and then maybe I would say like inhibits this reaction with the kind of arrow with the flat end of it. So in other words, I could say high levels of arginine would be proportional to dig, right? Because if egg causes reduction of dig, arginine, high amounts of it, would inhibit the reduction of dig, meaning that it would be proportional to dig. Does that kind of make sense?
Yeah.
Okay. Cool. All right.
All right. So an operon containing 2 genes, cardiac glycoside reductase 1 and 2, CGR1 and CGR2, was upregulated in Elenta cultured in the presence of digoxin.
All right.
So Operon, I would just be visualizing like basically like a straight line and then 2 genes within like that strand of
Yeah. So tell me what unique property, like what's unique about an operon?
so as a promoter region,
Do we have operons?
no, don't.
So why not? Or like what do we have instead? Or like what's the main difference between having an operon and not an operon?
I mean, we go through like translation,
Okay.
and then bacteria has operons where they have different genes.
So do you know the word like polycistronic, and do you know what I mean when I say that?
Yeah, so multiple
Yep.
genes. Yeah.
So operons typically, as you said, like typically contain more than one gene that could be transcribed and translated at the same time. But and that prokaryotes could do that, but we can't do that. Eukaryotes can't do that. We have monocistronic, RNA, so we can only make one product at a time, right? Now, but I like that you caught operon as a buzzword, and right away were defining it. Now it seems like the genes CGR1 and 2 are reductases, so maybe they do mean reduce in the redox context. So, I'm still not completely sure, but I think that if they're saying reductase, that's what that would do, right?
Yeah.
So perhaps they actually do mean reduction in terms of redox. And if that's the case, I would have to adjust what I wrote here. So let me do something like this. I'm gonna erase that. And I'm gonna write that egg is proportional to reduction of dig, and high arginine just inhibits that reaction, but also that egg would be proportional to CGR1, comma, CGR2, right? So now I have the presence of egg lenta causes more of the reduced digoxin, and high levels of arginine inhibit this reaction, and egg is proportional to the CGR1 and 2 reductase enzymes. Okay. All right.
Okay, so to test whether variation in E. lenta strains affects digoxin reduction. Okay, so it is reduction. It seems. Yeah. Fecal samples containing the gut flora of 20 healthy individuals were analyzed for digoxin reducing ability and a CGR ratio, specifically the abundance of the CGR operon relative to the E. lenta ribosomal RNA gene. These results divided the samples into 2 groups.
Okay, so what are they doing here?
So they're trying to figure out like what combinations of different G or different strains will I guess result in like a couple different things. So like reducing ability of digoxin and then the ratio of CGR.
Okay. So then we got table 1, and we see reduction and CGR ratio in fecal samples. Note asterisks indicates p-value less than 0.05 compared to group 2. So we have digoxin reduction, group 1 96.25%, group 2 12.82%, and the ratio with 1.05 for group 1 and 0.42 for group 2. Okay, so what I'm thinking, I mean, I don't know this for certain yet, so I'm not going to write it, but what I'm thinking is that there could be a relationship between the CGR ratio and digoxin reduction percent. They both seem, if like they go up, both of them seem to go up together. But yeah, that's pretty much that.
Would you make a note of these results like before even getting to the questions, or would you like come back to it?
Typically, I would try to decipher what something's telling me. But like for this one, that's the most, like what I said is the most I could really do. Like I don't see anything other than group 1 that has a higher CGR ratio, and then I mean it has like double or a little bit more than double the CGR ratio of group 2. However, it has like almost 10 times the digoxin reduction percent, compared to group 2. So I can't see anything that I can really get from this other than group 1, both of these things go up, and it's like, and it's not like super proportional either, but that's all I would see. Or that's all I would kind of take away from it. Now when we're doing passages together like this untimed, or when you do the passages on your own untimed, you also want to go through it slowly like this. But on test day, or when you start taking your practice tests, what I say is when you're reading the tables and charts and graphs and stuff, spend like 15 seconds. Spend 15 seconds to see if you can come with some type of understanding of what the graph or figure or whatever table is trying to show you. If it takes longer than 15 seconds, then you simply move on and go to the questions. I mean, move on, read the rest of the passage, go to the questions, and if a question asks you about it, about going to the table or something like that, then you can go back. So that's basically 2 extremely different ways. When you're doing it untimed, you do it slowly, you really want to understand everything every step of the way. But test day, everything under the time conditions, 15 seconds, no more than 15 seconds, because a lot of times either they'll ask you something about it that you can answer pretty easily, like it won't be too complicated, or they'll ask you something about it that will be complicated, but that'll still just be like one question. So, if you just remember that every passage is just like 6-ish questions total, then you can kind of see like overall cost benefit that it would be more worthwhile to only spend more time on the data if you need to. Yeah.
Okay. So next paragraph, the gut flora from group 1 samples reduced digoxin to a similar extent as Elenta, Elenta strain DSM, whereas group 2 was more similar to Elenta strain FAA. Group 2 fecal samples were then cultured in the presence of either the DSM or FAA strain. The ability of these mixed cultures to reduce digoxin was compared to the effects of the group 2 gut microbiota alone or each strain alone.
Okay. Yeah. So let's see what they're saying here. So they're saying that the gut flora, aka gut microbiota, reduced, or gut flora from group 1 reduced digoxin to a similar extent as the egg thing did, and the egg strain is the DSM thing. Whereas group 2 was more similar to this other type of egg strain FAA. So okay, we have 2 types. Okay, maybe I would write like group 1, maybe like associated with DSM, and then group 2 associated with FAA. All right, and then cultured in the presence of either that or that, and then the ability of these mixed cultures to reduce digoxin was compared to the effects of group 2 gut microbiota alone or each strain alone, figure 1. So okay, yeah, I would evaluate this graph here or figure. So but you always want to read this caption first. So it says percentage of digoxin reduction in group 2 gut microbiota samples co-cultured with the DSM egg lenta and FAA egg lenta, and the asterisk means the p-value is less than 0.05, and then double asterisk it's smaller than 0.001. And then for this like a bar graph like this, I would really just do it in a you always want to do something in a slow, systematic way. So just to minimize mistakes. So I would just start off with the leftmost bar, and and I see that there's a certain level of reduction percent, and I would just go from left to right, and then the next one is the FAA thing, and there's a significant reduction or significant drop in reduction percentage. And then we have gut microbiota with a low level, similar to FAA. And then gut microbiota plus the DSM goes up, and it goes up higher than DSM on its own. So that's good to know. And then last, we have gut microbiota plus the FAA, and it's like a really tiny amount. So yeah, so it seems like so okay, what can we, I guess, take away from this?
I'd say, I mean, DSM definitely has a huge significance on the reduction ability. And FAA doesn't seem as applicable in both like the E-lenta and the gut microbiota.
All right. So um okay, so it seems like if we like okay, the comparisons I would make would be DSM on its own, and then if you add gut microbiota, it increases the amount of reduction, right? So over here, where I wrote gut microbiota is proportional to dihydrodigoxin, which is proportional or inversely proportional to digoxin, I'm going to add proportional to reduction percentage, right? Because when we have it alone, we have a certain amount, but when we have it with gut microbiota, the amount of reduction goes up. So I'll say gut microbiota is proportional to reduction. Now, then the FAA on its own is pretty low, and the gut microbiota is pretty low. But when you have gut microbiota and FAA, it's still pretty low. So, I maybe would then in parentheses here write down DSM, because gut microbiota is proportional to the reduction percent for DSM, right? Because we see it going up here. But I can't say that gut microbiota is proportional to reduction percent in general, because if it was, this should go up.
Right.
That's why I put in parentheses DSM. So yeah, does that make sense?
Yeah, it does. And it also, I mean, the way that you outline everything really like minimizes the like clutter that I find myself.
Yeah, that can, like, with highlighting a lot, like that can, and just these BB passages in general, like there's just so many acronyms, right? There's DSM, there's FAA, there's ATPase, uh there's just a ton of stuff here. So yeah, if we now take a look at everything that we wrote that digoxin is inversely proportional to that ATPase, which in other words mean that it can, inhibit that ATPase. We know that gut microbiota turns digoxin into dihydrodigoxin, which means that it's going to be less of the actual digoxin. I didn't write it, but maybe in hindsight, I would put in parentheses for dihydrodigoxin that it's an inactive form of digoxin. But that's why also it's inversely proportional to digoxin. We also have for egg that it produces more reduced digoxin, and that it also causes the CGR1 and CGR2 reductase enzymes to be formed. And high amounts of arginine can inhibit the egg, causing this reduction reaction. And then I wrote down like how group 1 is similar to DSM and group 2 is similar to FAA. And then lastly, just the gut microbiota proportional to reduction percent for DSM. But that should cover everything. So that's what I would write when doing passages like this.
Okay.
So yeah, so you could take a screen, or I could take one for you and then put it in the chat. Yeah, here we go.
Good.
Mm-hmm.
Okay. So first question, the CGR1 and CGR2 proteins are produced from, so I guess going back to my comment about like the Operon, how so it would essentially just be lying on um, like one strand. So that's something that I would like be visualizing. I'd probably like draw it out. So first option is separate mRNAs transcribed from different promoter sequences upstream of each gene. Okay, so this wouldn't be correct because we're not talking about different mRNA strands. We're talking about just one.
What would A describe?
It sounds like um I mean, sounds like transcription,
So
polymerase.
remember what was the difference between
There's
prokaryotes?
like trans
What's up?
difference.
Oh yeah, between prokaryotes and eukaryotes in that.
Difference being like the fact that eukaryotes are monocistronic and
Yes. So if eukaryotes and monocistronic, you would need each gene would need its own promoter sequence, right?
Mhm.
So A is describing eukaryotic transcription, right?
Mhm.
So good. And you see how you recognize the buzzword Operon. And right here, they're asking us about it. So good job on that. But yeah, A would be what happens in a eukaryote, right? So okay, so we can get rid of that.
B single mRNA transcribed from a single promoter sequence upstream of the operon. That sounds about right.
Mm-hmm.
Yeah. I think I'd probably just choose that and move on.
Well, you still want to look at every choice because you might get something better.
Fair enough.
Mm-hmm.
Then C, a single RNA transcribed from a single promoter sequence and alternately spliced to produce separate mRNAs. I wouldn't choose this one because alternate splicing, I mean, that's a post-transcriptional modification, but that doesn't really seem within the scope of this question.
Well, I don't know about it being wrong because of scope, but so what else could it be wrong because of? Oh, well, what is alternative splicing?
You're basically like splicing out different exons and like different patterns essentially to get different genes.
When you say slicing out, do you say slicing out exons or
Or like basically transcribing them in like a different pattern.
so? But tell me more. Tell me about the exons.
The exons contain the genetic information.
Okay. So what else is there?
What else is there within like
Yeah,
like
like you have exons, and then what else do you have?
introns as well?
Yep. And then what happens with those?
Introns are taken out.
Yep. Yeah. So if you have like let's say exon 1, some introns, exon 2, exon 3, exon 4, you can get let's say exon 1, exon 3, exon 4. You can get exon 2, exon 4. Like there's many combinations of those exons.
Mhm.
Well, splicing itself means you're removing the introns and you're splicing together the exons. And alternative splicing is when you have different order, variants of those exons.
Mhm.
Now, as you said, that's a post-transcriptional modification. Um, And one of the things about prokaryotes is, in addition to them, you know, being polycistronic, they also have the ability to transcribe and translate at the same time. So if you can transcribe and translate at the same time, there is no post-transcriptional period for there to be for there to be something like a modification. So C is specifically describing eukaryotes, right? And there's also kind of like a hint where, we have separate mRNAs, and then compared to like, yeah, well, I guess we do have a separate mRNA here, but we at least have Operon and B and C. I'm sorry, B and D.
Yeah.
Um, but Oh yeah, and then so why would D be wrong?
Mm, I mean, we wouldn't be transcribing from different strands to begin with, so I think that would probably be my biggest giveaway.
Yeah, right. You have separate mRNAs, which again means that they're monocistronic.
Yeah, and I guess it says translated, so
Mm-hmm.
yeah.
Yep. So yeah, good job. Answer should be B.
Yeah, I think the first time that I took it during my practice exam, I put C. And then when I redid it without looking at the answers and then like actually drew out the Operon, it like B was just obvious to me.
Mhm.
It's- yeah,
Yep.
but the didn't. Okay, so which actions could contribute to the positive inotropic effect of digoxin on cardiac myocytes? Okay, contribute. So basically, what could contribute to the increases in the force of contraction? Okay. Decrease transport of calcium to the extracellular environment. That makes sense because, I mean, if we aren't able to take out like the calcium that um that is now in like the or that is now interacting with the troponin and tropomyosin interaction, then it makes sense that this would increase that. Increase availability of intracellular. I mean, that makes sense because if it's more able to bind, then it would increase contraction. And then increase overall calcium stores. That just means that there's going to be more calcium coming from the sarcoplasmic reticulum. So D, I would put D.
Yep. So yeah, the calcium can function in the, you mentioned something about the troponin thing.
Yeah.
Um, so it would like bind to it and expose a, an area that can bind to the in which the myosin and actin can bind to one another. But yeah, I think it'll be the
Which type of membrane transport is directly affected by cardiac? Okay, directly affected.
so remember when we saw, so what buzzword did we see that made us already think of this type of question?
The ATPase.
Yep. So you see how what we're, you know, what we're now kind of seeing, kind of like the word buzzwords that we're seeing, like they're asking us questions on.
Yeah.
So that's just shows how useful that is because it's stuff that they're literally asking questions on. Yep.
Yeah, the first time I put secondary active transport because I think even going back to when I was first breaking it down, I was thinking of like the ATP pump.
Yep.
Um yeah, which creates that like proton gradient. So I was like, oh maybe, but
Yeah. Like yeah, secondary would be like when it's coupled to something like an unfavorable reaction that's coupled to a favorable one that can kind of give it the energy, but at least we can get rid of like the diffusion ones, right?
yeah.
Yeah. And then ATPase, like it's requiring the breakdown of ATP like straight up. So uh that's it's not going to be coupled because it's just straight up using it. Yep.
Okay, so CGR2 expression in Elenta exposed to digoxin is shown. Note L indicates low and H indicates high. Dihydrodigoxin levels in the culture medium of Elenta cells would most likely be highest in which data set?
Okay, so yeah, this type of question is like one of the worst because they like in the question stem itself, we need to do some data interpretation. So let's try to interpret what this is showing us. And again, I would just start from like left to right in a slow and systematic way. So yeah, how would you kind of describe it?
Okay, so I mean, I'm looking at exponential growth and stationary phase. So I see like this is like a completely different group as opposed to this one.
Tell me
Uh,
what the like why there is exponential growth and then stationary phase. What does that mean?
Exponential growth being that, I mean, we're just saying like, um, what even explain? I mean, I guess like, it's like the proportion is squared essentially. So like, we would be saying like, I can't remember, this is not a parabola. It's like, I can't remember what the specific.
Yeah, so bacteria can create more of itself through binary fission, right? Like if you have let's say this bacteria, it will divide into two. So each time it divides, you're doubling it. So it'll be not necessarily the squared term, but it'll be 2 to the power of n, and n is the number of, divisions there are. But that's what gives you that, doubles each time, exponential growth period until enough of the like either like all there's no more room or there's no more like food which would cause it to reach its carrying capacity and then stabilize.
Yeah, which would be the stationary phase.
Good. And then all right, and then we see so CGR2 expression as the y-axis, and then we have, and I'm just going to start at the leftmost thing. So we have arginine amount low, digoxin negative, so not present, and CGR2 expression is 0. Um, okay, and then I would just look at the next one, and then its arginine amount is still low, but now digoxin is present. And guess what? We have a ton of CGR2 expression. Um, and that's something that, okay, so I guess, okay, so we see that. And then after that, we have this one where arginine amount is high. And when digoxin is not present, we get 0 CGR2 expression. And then arginine levels high and digoxin is present. We have, something that has some CGR expression, so it's more than, 0, but it's not as high as the condition when you had digoxin with low amounts of arginine, right? So, okay. And then let's see, the one after that, stationary phase, low arginine, no digoxin, 0 yet again. So that's exactly what was, in the data set 1. And then data set 6, low arginine, presence of digoxin, and that seems to be this middle number over here. And then no digoxin for data set 7, so that's 0. And then data set 8, high arginine and presence of digoxin, and it's a small amount here. So it seems like in the presence, well, it seems like the CGR2 expression can go up. Let's see how I can put this. Maybe like digoxin proportional to CGR2, and arginine can inhibit that, the digoxin causing the CGR2 to be expressed. Yeah, I think that's what it's saying. What do you take away from it?
Yeah, I mean, as long as digoxin is present, like arginine is not going to be able to completely inhibit CGR2. So,
Oh yeah, that's actually that's a good way of putting it too. Like if you don't have digoxin, you won't have CGR2 at all. And then it's just a matter of having it and then having it with low amounts of arginine and then having it with high amounts and comparing, okay, low to high, like it goes down. And then here low to high, it goes down as well. So sometimes I also write like kind of like I call them like greater than hierarchies, like arginine high plus digoxin is less than, I should have started with low. So I'll just do that. Low, and then actually this will be greater than sign. Arginine high plus digoxin, and then yeah, pretty much that for both exponential growth and stationary phases. So, all right, cool. So let's see. They're asking, okay, where would the dihydrodigoxin levels be highest? So we want to look at what we wrote in, in our relationships to see, yeah. So yeah, what are your thoughts?
so is this two separate? Okay, no, this is okay. This is dihydro, but I mean, we're not necessarily. We're not necessarily comparing it to. Well, actually, I guess we are because we're looking at looking at bacteria. So, okay, so basically, dihydrotoxin would be highest when E-lentes.
Okay, so okay, I think I can add something to what I wrote so far. Because now that I think that reduction really means like redox reduction,
Mhm.
I'm pretty certain that's what the dihydrodigoxin is. It's the reduced form of digoxin. right?
Okay, I see.
So here I wrote egg proportional to reduced form of digoxin, and then also proportional to CGR1 and CGR2, right? So now this reduced digoxin that I wrote is the same thing as dihydrodigoxin. Right?
Yeah.
So we want to see okay, so now we're equating the dihydrodigoxin to the reduced form of digoxin. So if we're looking for the highest dihydrodigoxin levels, we should be looking for some condition where we have the highest amount of reduction of the digoxin, and what can possibly do the reduction. I'm going to guess these reductases, right? CGR1 and CGR2, right? So
then the more CGR2 we have, the more dihydroxin.
yeah,
Okay. So in that case, I would choose 2 because it has the highest.
yeah. 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 So that's like, so most of this passage we kind of understand, but you know, just that thing with reduction, that's kind of annoying. Because like because it can mean less or it can mean redox reduction, but we ended up coming to the conclusion that it's redox reduction. I think it was when I saw the reductase being formed, that's what made me make sure that it was redox reduction.
Mhm.
And then from there, it's just a matter of like equating terms. Because when I write proportionalities, like it could kind of work with an equal sign as well. Like, you know, if you know, yeah, uh, meaning reduction, like that's where I wrote reduction digoxin is equal to the dihydrodigoxin.
Mhm.
But yeah, this type of question is hard because you know it involves data interpretation itself. Um, and there's a lot going on. It's not like a regular X, you know, axis, Y axis graph in BB. It's like X axis has like arginine levels, digoxin levels that are different, and then Y axis has just one, but then there's also like an exponential phase and stationary phase, and you know, it can kind of lead you to like down a rabbit hole if you think too much about these, but essentially yeah, we just want to look for the relationship in this case between CGR2 and dihydrodigoxin, aka reduced digoxin. Mhm.
Really kind of comes down to like how well you organize, I guess, like the portions too.
Yep, yep. Yeah, 'cause
So
it becomes like a logic thing each kind of step of the way.
yeah,
Mhm.
makes way more.
Yep. Yeah, it works for a lot of stuff in life. It's like, you know, it's like if you're investing in stocks, you look for something that would be like an inverse proportion to stocks, which is usually like gold or crypto or something like that. And you know, if those things go up when stock prices go down, or like treasury yields or something, like it's always useful, like, you know, no matter what you're doing, to look at those types of proportionalities, which by the way could also be like in psych soc, instead of calling it inverse proportion, they call it a negative correlation.
Yeah.
And then for a direct proportion in psych soc, it would be a positive correlation. But yeah.
Okay, so in psych social, do you like draw out portions in the same way? So
Yeah, I use like every opportunity I can to write proportions out. Um, so yeah, it might be there might be like psychosocial like other kind of things that you have to be aware of, but for BB I think is where the proportions help the most, just because there's so much to work with, so much to keep track of.
Mhm.
But we have another question on this buzzword right here that we already talked about, right? So you see right away that at least so far, I don't know what 23 looks like, but so far, this table, we haven't had to use it, right? So, so that's why what I said about the 15 seconds is a good strategy, because otherwise, you'll spend too much time on the table and they might either, again, I haven't seen 23, but they'll either not ask you about it so far, or they'll ask you about it in a way that maybe you can answer without knowing every little detail.
Mhm.
But I hope that you do see that so far we've been being asked about at least, 3 times, I think, about things that we know that we already identified as buzzwords. Yeah.
Gotcha. I mean, this is pretty straightforward. It's just whether or not you know
Okay.
it. Okay,
All right.
20.
Yeah, I think this is it then. This is the last. So I think this would be talking about table one then. But but yeah, go on.
Which statement is least supported by the experimental data? Okay, so A is a high CGR ratio correlates with increased excretion of reduced digoxin. So high CGR ratio. I remember it saying like the higher the CGR, the less Elenta, I think. Let me make sure. Okay, relative to Elenta, yeah. Okay, so high CGR correlates with increased excretion of reduced digoxin. And so then in that case, and we have to go to our proportionalities and think, okay, well, if we're having high CGR, then we have less Elenta. What does that mean for digoxin?
So the stuff in the experiment, like, I guess this is a good time to say this. Like, it's stuff in the experiment
Oh,
does does not, yeah, does not need to be consistent with what we've already written out, because what we've already written out is like theoretical, like the way that things should behave. But whenever you have experimental, results, it could be whatever it is. It doesn't have to be consistent with what we have in theory. But um but what we do see in table one, though, is that there is, I'll use green, there is a proportion, seems to be, between CGR ratio and digoxin reduction, right?
yeah.
Like that was like, remember how I was like, you know, like when we looked at this and it was like, okay, the only thing that we can really see is that group one has a higher amount of CGR ratio and a higher amount of digoxin reduction. So we could reasonably say that these are proportional to one another. If you have a high CGR ratio, you should have a high digoxin amount that's reduced, right?
Yeah.
So, and yeah, when you see that experimental data, yeah, that's what it's asking us to look at. So, so would A be true or false?
This would be true, but I mean, they're looking at least supported, so we would
Good.
just say. The CGR operon is functionally more active in the FAA strain than in the DSM strain. I know that's not supported because I remember like even looking at the figure, anything with FA just had significantly less activity. Choose that one, but just for
So is B true or false?
yeah, this is false. I would say B.
Okay.
But for the sake of going through it, we can also go through C and D. A low CGR ratio correlates with reduced digoxin inactivation. So low CGR ratio here, reduced activation. So this is true because I mean we can see that it significantly reduces the reduction percentage.
Yeah, that's funny because we have the word reduced again, but this time they mean less. But
Yeah.
that's annoying. But yeah, that's true.
The abundance of the CGR operon and gut flora may predict the bioavailability of digoxin in individual patients. I mean, I feel like that's kind of it seems out of the scope-ish from like the two experiments. The abundance of the CGR operon. I mean, I suppose like we could say that the more CGR ratio, or the higher the CGR ratio, the higher abundance of CGR operon to some extent.
Okay.
And then, yeah, I mean, I guess because it has it's significantly um, statistically significant in this sense, then we would say that that's like true.
Mhm. Yep.
Okay. Yeah, I'm gonna practice those proportionalities because I think that's pretty huge in terms of like organizing.
Mm-hmm.
So, yeah.
Yep. Yeah, and also the buzzwords, right? Because that's exactly what we saw here, right? If you go back to 18 for a sec. So 18, so we saw the buzzword operon, and then we talked about how that works, and it works because with polycistronic stuff. So B right away we were able to answer because of the operon buzzword, and then you go to 19. 19, we we could also kind of answer because we were well, I guess this is less of a direct thing that we were able to think about from the buzzword, but we do know about well, I guess this one we could say isn't related to a buzzword, like that's fine. Um, you can, yeah, go to the next one. And then this one, definitely, we talked about it with the buzzword sodium-potassium ATPase. All right, and then the next one. Then this one, not buzzword. And then this one, buzzword. Same thing, sodium-potassium ATPase. And then then the last one was just the table. So 3 out of the, what, 6 here, or 5, 18, 19, 20, 21, 22, 23, 6, 3. So half of those were stuff that we were expecting from buzzwords.
Yeah.
So yeah. So yeah. Buzzwords definitely help. And yeah, the proportionalities as well. So yeah, definitely as you move forward, the um that will help, yeah.
Okay. Sounds good then. Yeah, that was kind of the main one that I wanted to go over because I think that was probably the most confusing-ish just because of how much
Mm-hmm.
packed into it. Yeah, the other ones I could figure out pretty.
Okay.
Yeah. Yeah. That's all I got for bio. Yeah. I think I mean, I'm gonna take my next practice exam.
Yeah,
Like,
so when did you take this exam?
um, took it last Monday. I think Monday, yeah.
Oh, so tell me, okay, because I want to keep track of your full, like how you're doing on your full lengths. Did I not make a spreadsheet for you? Okay, I guess not. Yeah, tell me, so you said you took this past Monday. How did what was your score and your breakdown?
It was 500. Chem Phys was 125.
Okay.
Cars was 124. Biochem was 125.
Yep.
Psychosocial was 126.
And the previous full lengths that you did were which ones? I probably wrote it down on one of my other notebooks.
I don't think we went over my um other scores.
So so okay, so this one so yeah. And then which other ones did you do? The
I took a few Kaplan ones.
okay, did you take any other AAMC one?
Yeah, I took let me see. I took AAMC 2. Let me see what my breakdown is. I think that was a 495, but let me just double check. But yeah, I mean, for the longest time, I just could not get cars down, but I think I finally have it. So I'm expecting my next score to like actually, because like cars are just bringing my score down so much. But um,
Oh, it's I found where I wrote, well, wait, did you take AMC full length twice? I'm sorry,
mm,
AMC full length three twice?
no, I only took 3 once.
Okay, because I wrote from the 19th a 499 for that, but I guess it's really the 500.
Let me double check. Let me double check. I was just saying that from. Okay, yeah, no, it was 499. I think. Okay, so my cars was 123, not 124. Yeah.
Okay. And this was when we
So I took that. So I took both before. Actually, okay, so I took number 2 before we met for the first time, and then I took 3. I think the day after our first session, maybe 2 days after. And my bio, because we only did bio that day. My bio went up about a point or two, and just double. Yeah, so my number 2 was 495.
okay.
Yeah.
So um when was your test again?
Thursday.
Oh crap. On the
It's Thursday, 27th, yeah.
yeah, okay. So I'm guessing you're like, are you gonna take one more test?
Yeah, just one more test, yeah.
And I'm guessing it'll be AMC full length 4.
Yeah.
Okay. And did I talk about timing checkpoints with you?
Yeah, you did. Hour, um, I should be through 20 and then 45, 30.
Yeah. So the idea is not that when you have an hour to look what number you're on. The idea is look what number you're on, and if it's number 20, you look at the clock because I don't want you to be looking at the clock constantly, right? So when you get up to question 20, you look at the clock. It should say you have an hour left. You know, if you have less than an hour left, gently tell yourself to go pick up the pace a little bit, but just gently. I don't want you to rush. Um, and then once you're on question 30, check the clock. You should have 45 minutes left. And again, if it's less than that, go a little faster. And then when you're on question 40, you should have 30 minutes left. And ideally, you want to have some time. Oh, and then also don't let any question take longer than a minute and a half. Obviously, you're not going to be specific, like looking at the clock to see if you're really spending that long or not. But if it feels like you're spending a lot of like more time than than that, just make an educated guess, and flag and move on. That's true for like calculation questions too, because that could be really easy to kind of tell yourself, oh, okay, I know I know how to do it. I'm just, I didn't get the right answer the first time, so let me check my math, let me do it again. That kind of can really be a time sink. So for calculation problems, I would say do it once. If you're not getting an answer after your first try, guess, flag, move on. And if you follow this with discipline, you should have some time left over at the very end. And it's better, like, let's say the alternative, you're spending 10 minutes a passage and and you have two passages left. But the second to last passage, let's say, takes longer than 10 minutes, then you'll have less than 10 minutes for the last passage, and that could work against you. But if you do it in the way that I'm describing it, then that means that, like, ideally, you'll finish the last passage, the last question, with, let's say, 3-4 minutes left. It's better to be in that position because in that position, you will have seen all of the passages and all of the questions. So when you have that time at the very end, you just hit, review flagged, and you use your own judgment on and your appraisal of, like, this question that I flagged, I'm 50% certain of. This question I flagged, I'm 25% certain of. This one, I'm 30% certain of. So when you go back to review the flagged ones, you'll do the ones that, you have a low appraisal of your answer being right. Unless, of course, it's a very time-consuming question. But yeah, I would incorporate that type of timing strategy for you. As well as meaning the checkpoints, as well as the 1.5 minute limit, and just getting into the habit of, if you're not 100% certain of something, guess, make an educated guess, flag, move on. Hopefully you'll have time at the end to review flagged questions. And yeah, basically that. Oh, and then I went over blind reviews with you too, right?
Yeah.
Yeah. So yeah, after your full length, like be done, after that full length, like on the same day. Don't do it on the same day, but the next day after your full length, you could do your blind review and review.
Gotcha.
And yeah. So when are you doing that, your next test? Tomorrow or something?
I think I'm gonna do it. I think Monday, or maybe I should do it. What do you think? Tomorrow or Monday?
Yeah, I'd say tomorrow, because like I would preferably want my students to take their last full length a week before their actual exam. And you know, that would be, if you take it on Monday, that means on Tuesday, maybe you do your review and stuff, and then we meet after that. But I think that's cutting it too close. I would say to take it at your next earliest convenience. So that if that's tomorrow, then yeah, I would say take it tomorrow and then rest for the rest of the day. And then Monday, do your blind review and review of it. And then let's meet on Tuesday or something,
Okay.
and we can go over stuff from that exam. And then on Wednesday, which is the day before your exam, don't do anything, any other kind of questions or passages. Just kind of like chill out a little bit, like do passive review like Anki and stuff, but kind of give your mind a chance to refresh um for Wednesday, and go to bed like, because everyone has a hard time going to bed the night before their exam. So if you go to bed like 2 hours earlier than normal, or than what you usually do, that can at least account for that. So yeah, I would say take your test tomorrow, then rest, and then Monday do your blind review and review, and then Tuesday let's meet, and then Wednesday just passive like review like Anki, um, and stuff.
Okay.
Okay, so
Tomorrow's gonna be my high score.
so yeah, I mean, like, you're no matter what, like, no matter what you're getting, like, you're still learning. Like things that you learn today may not come up for tomorrow's test. So, don't put too much weight on it. But yeah, you should be somewhere in the ballpark. So okay, let's see. On Tuesday, are you available like earlier, like let's say like 2 p.m. Eastern
Yeah.
on Tuesday, the 25th? Okay, so put you in. So I put you in for Tuesday, the 25th at 2 p.m. Eastern.
Also, when I was